Welcome everyone. This is the part 2 of the complement system by Zohib Tahir. So in this video, we will be discussing two pathways of the complement system. Number one is the classical pathway and number two is the alternate or alternative pathway. So let's get started. So we have a classical pathway. We have a couple of points there, but before explaining these couple of points, let me show you a diagram first. Here is a diagram. Here you can see bacteria, and more specifically it is gram negative. Why it is gram negative? The question is this. So let me tell you something about gram negative. In gram positive bacteria, we have a cell membrane, and around it we will see a thick layer of peptidoglycans. Okay, and this is our gram positive. Okay, if I draw the common or the general structure of gram negative bacteria, then it has a cell wall. Okay, then it has a thin layer of peptidoglycans around the cell wall. Then we have outer membrane. Okay, this is an outer membrane, and this is the structure of gram negative. Okay, now in case of gram negative bacteria, we can see cell membrane over it, which can be drilled by MAC very easily, rather than breaking into the cell by breaking into the peptidoglycan layer. Okay, so easy way to perforate or to opsonize or to phagocytize the bacteria, gram negative bacteria, is the complement system. Okay, so let's go back to the first slide. Okay, antibodies antibody binds to C1q in the presence of the calcium ions. Okay, antibody binds to C1q. Which specific antibody must bind to it? This is the question. So let me tell you that IgM and IgG, these two are the antibodies which will bind to the C1q, one of the subunit of the C1 component of the complement system. C1q will cause the activation of C1R, and C1R will cause the activation of C1S. Okay, here you can see C1q bind to antibody, this one and this one. Let me tell you another thing very interesting. If there is an antibody, let me draw it only in this fashion. This is an antibody, and over the antibody there is a specific part with which C1q will bind. Okay, this active part is not much active when it is not binded to the surface of the bacteria. When it is bound to the surface of the bacteria, this part will bulge out. Okay, when this part will bulge out, what will happen? C1q will bind to it. Okay. Okay, C1Q will activate C1R and C1R will activate C1S. In the next slide, sorry, in the next slide, you can see that when all the components of the C1 have been bounded, then comes C4. Okay, you can see here C4, C4A and C4B. Actually, each component has two parts. Let me draw it in this way. This is part 1, this is part 2. One part is known as the A part, and one part and second part is the B part. Okay, so it is inactive state. Okay, it is inactive state. When it has to be activated, what will happen? A will be shifted over there and B will be bounded to the previous component. Okay, let's go back. Now C1 attaches to C4B, or I must say C1 causes the activation of the C4B. Here you can see C4B has bounded to the C1Q, and on the surface of the bacteria, and C4A, here is the C4A, it has been detached from the C4B, and now it is present in the plasma. Okay, these all apples have some role that will be discussed in the clinical significance and the biological effects. Then C4B will activate C2, and again here is 2B, and it will leave its apple part to A, C2A. This is C2B. Okay, then comes C3B. C2B will cause the activation of the C3 and will cause the detachment of the C3A. So C3B will bind to C2B. Now C3B will cause the activation of C5, and C5 will detach its A part, that is C5A, and C5B will attach to the C3B. Now C5 will activate C6, C7, C8 and C9. Now C5, C6, C7, C8 and C9, these all are responsible for the for the phagocytosis, and these all will be called as MAC. And what is MAC? MAC means membrane attack complex. Membrane attack complex. Okay, let's go next now. Sorry. Okay, this is one of the important slide. First of all, we had C1Q binding to the antibodies. Okay, antibodies are binding to the antigenic site here. Okay, now this C1Q will cause the activation of the C4, and C4B will bind to C1Q. With C4B to B will, there is a mistake. There is a mistake, and solid mistake. This one is C2A, not C2B, and this one is C2B, not C2A. Okay, this is a mistake in the diagram. C2b will be further attached to C3b, and C3b will cause the activation of the C5b. C5b will attach itself with C6, C7, C8, and C9. These C5, C6, C7, C8, and C9, these will be arranged in the circular pattern like that. Okay, and this is actually known as MSE here. And these all the components will drill the membrane of the bacteria, will cause the perforation in the bacterial cell membrane, and will cause the destruction of the bacteria. Let's go next. I have told you before that C5, C6, C7, C8, and C9 are known as MSC, means membrane attack complex. Okay, let's go next. Opsonization. What is opsonization? Video is getting very lengthy. Opsonization. Let me define opsonin first. Opsonin is the any molecule that enhances the phagocytosis. What is phagocytosis? The act of eating up the antigen, or the foreign body, or the bacteria, virus, fungi, whatever it is. By marking an antigen for an immune response, means uh, whatever we have, the components of the complement system, all the components, not all the components, but some of the components. So for example, C3b, C4b, and C1q, these all will cause the activation of the immune system, will provoke our immune system, and will cause the activation of immune system against that antigen with which they are bounded, and will attract most importantly C3b will cause the attraction of the neutrophils. Okay, we will discuss it later, and we'll come to the third and the last part of the complement system. We will discuss it. Okay, so there are some of the components which will attract the neutrophils towards the antigen, which will cause the production of the antibody C3a. Okay, some of the components present in the plasma, a parts. Okay, these a parts will cause the production of the will cause the attraction of the neutrophils, and some of the components C3b and C5a, or any other component, will cause the production of the antibodies. Okay, most importantly, C3b will bind to the activated B cells, and activated B cells will cause the production of the antibodies. Rather, I must say C3b will cause the stimulation or the activation of the B cells, and B cells in result will produce the antibodies. So opsonin is a molecule, is a compound which will enhance the phagocytosis by marking an antigen for an immune response, or marking dead cells for recycling. Then we have, and the process of facilitating means there was C3b, there's C5a, C1q, C4b, and some of other components. These all the components will provoke a phenomena, okay, in order to attract neutrophils and the macrophages towards the antigen, to kill the antigen, to phagocytize the antigen. Such a process is known as the opsonization. In case of the classical pathway, we have seen C3b, C4b, and C1q as the opsonins. Okay, I have told you in the first slide that IgM and IgG, these are the two antibodies which will start the classical pathway. So it was the classical pathway. Now it's time to discuss alternate pathway that is very short. Okay, this time to discuss alternate pathway, and we have only mechanism of it, and then we will see the difference between alternate pathway and the classical pathway. So we have the mechanism. In the mechanism, we will see that if we have the bacteria, in the next slide we have the bacteria. If we have the bacteria, you can see the bacteria here, and on the bacteria we can see endotoxins. We have endotoxins are more in gram negative again, but where will the gram positive go? What about the opsonization of the gram positive? So we will discuss it in lectin pathway, that how gram positive bacteria will be phagocytized, will be opsonized. Okay, so bear with us. In the mechanism, we will see that C3b will directly bind to endotoxin. Okay, here is uh, endotoxin surface molecule with which C3b will bind, and C3b will cause the activation of special factor known as factor B, or simply B. If I go back and tell you something, here is part B and here is part A. Okay, and total is B. Okay, now then, C3b will cause the activation of the factor B by removing this apple. So we will not have this apple there. Okay, so B, we will get what we will get, we will get B, B, and in the serum or in the plasma we will get the A part of it, B, A. Okay, now B and small b activates another C3. So C3 will be activated in such a way that its A part will go away, its apple will go away, and C3b will bind to the B. Okay, then we have C. Yeah, I have to tell you about the convertase as well, so bear with us. C3b, then we have had B and small b, then again C3b. This whole will act to activate C5. That is why it is known as C5 convertase. Okay, now C5b will be attached to the C3b, and A, or apple portion of this C5 will drift away. So C5b will again form MAC, means membrane attack complex, and we will have the opsonization again. Okay, antibodies are not involved in this case. Then we have the, yeah, let me explain from this diagram. We had endotoxin there with which C3b bounded and its apple went away. Okay, then we had B factor bounded to the C3b, BA went away, then BB activated C3b and A part went away, then C3b activated C5, its A part went away, and C5b attaches C6, C7, C8, C9 and C8 form the MSC and causes the proliferation of the bacteria and killing of the bacteria. Okay, then comes the difference between the classical pathway and the alternate pathway. In the classical pathway, we will see that it is actually a type of acquired immunity. Okay, acquired immunity, while alternate pathway is innate. What do you mean by acquired immunity? We had antibodies, okay, and antibodies are acquired, are formed as a result of the exposure to the bacteria. While in case of alternate pathway, there was no antibody involved, so that is why it is innate. When we will have the first exposure of the antigens, then antibodies will be formed against that antigen. So we are acquiring it with the passage of time when the life passes by. Okay, now initiation. We have seen that in the classical pathway we had antibody binding to the antigen, but in this case there was a direct bond between C3b and the endotoxin. So this is one of the difference. Then we have role of antibodies. So antibodies actually are very important in case of the classical pathway, but in case of alternate pathway there is no role of the antibody whatsoever. So in case of classical pathway we needed antibodies for the activation of the whole process of the whole complement system. Okay, then we have the role of properdine. So properdine is needed for the activation of C3. We will discuss it later. And C3 convertase, yes. Now comes convertase. So C3 convertase means such a complex, which must activate C3. So in case of classical pathway, there was first C1, then we had C4B, okay, and after C4 there was C2B. So C1 causes the activation of C4B, so it will not be included in it. Rather, we will have C4B and C2B as a convertase known as the C3 convertase. Okay. But in case of alternate pathway, what we had actually, we had C3B directly bounded to endotoxin. Then we had B and its P part. These two things are the complex of C3B and BB will do the function as a convertase for C3, or I must say will function as a C3 convertase. Then we have C5 convertase. In C5 convertase, in case of the classical pathway, we had C4, C2B, C4B, C2B as C3 convertase. Then C3B will bind to the C4B and C2B and will act as a C5 convertase. While in case of it, C3B, C3B and BB we had act as C3 convertase. So again C3B will bind to the BB and will function as a C5 convertase. Then the components involved are the involved components in the complement system in the following pathway. In the classical pathway we have C1 to C9 all the components, but in case of alternate pathway we will have factor B, factor D, properdine, C3, and C5, C6, C7, C8, and C9. These are required in case of the alternate pathway. In If we talk about MAC, then there is no change, or there is no difference whatsoever if we see both the classical and alternate pathway. So this was today's topic. Hope you have enjoyed it. Thank you very much.